Hello students, how are you? I am really missing you all. Hope you all are good and doing well, and hope you are studying also very good because I am seeing all your messages in the group, and I'm very happy to see you active in the group. And many students, they're asking doubts even late night also. I'm really sorry that I'm not attending because you know very well I can't be able to answer very late doubts. Even some students are asking at 1:30. Even I've seen yesterday, if I'm correct, or day before yesterday, I exactly don't remember, they're asking doubts even morning around 4:30 also. And I'm very happy. Why? The other students are helping them. When I am not helping them, some other students are helping them, and I really appreciate those who are helping your friends, because sharing obviously doubles the knowledge. So it helps you also, and of course it helps your friends also. So sharing knowledge, I truly believe it, and you are doing very good job, and keep it up, my students. And I'm very grateful to you, those who are sharing their knowledge, those who are helping the other students. Now let us start our today's concept, that is microsporogenesis. And microsporogenesis, in the name itself it is clearly mentioned, microspore genesis. Genesis means what? To form. And microspore means this pollen grain, it is also called as microspore because it is of very minute size. That's why microspore genesis, microsporogenesis, that means the formation of pollen grains. How? Let us see. For that only we are making this video. Now, before we start how the microsporogenesis, that means pollen grain, it is formed, first of all how the stamen is formed we will understand, and slowly we'll enter into the concept. Now students, all these are points in a theory form, but you need not read all these things. Just to make you very easy, I made this flow chart. Very easily I'll explain, because anyway it is revision and most of you know all these points, but just like a part of revision I'm explaining this video. Now, this is thalamus. Hope you know what is thalamus. The stalk of the flower, it is called as a pedicel, and on the pedicel you can be able to see one elevated structure that is called as thalamus, and of course thalamus, it is also called as a receptacle. Whereas now, thalamus is a portion or a part of the flower where the rest of the I'm really sorry, I made a mistake. The rest of the flower parts, they are present. What are the rest of the flower parts? One, calyx; two, corolla; three, androecium; and the fourth one is gynoecium. All these particular four parts, they are present on the thalamus. Now, such a thalamus, if it wanted to produce a stamen, that means androecium means all the stamens together called as androecium, and every single unit of this androecium, it is called as a stamen. Where when it wanted to produce stamen, the thalamus, it produces a very small outgrowth. A very small outgrowth, it is formed on thalamus, and that small outgrowth which is formed on the thalamus, it is called as a microsporophyll primordium. Now this microsporophyll primordium, it is consisting of the meristematic tissue, and this meristematic tissue, it undergoes differentiation. By the differentiation, the rapid division takes place, which results in the formation of the stamen. And this stamen, it consisting of two parts. The first one, it is called as filament, which is the stalk, whereas anther is a fertile part, which is a bilobed and tetrathecous structure. Now, this is stamen. I'm talking about immature stamen, just now formed stamen. Hope you know what is the meaning of immature stamen. Immature stamen means this is just now formed stamen, which is not having the capability or ability to produce the pollen grains. I can say young stamen. This young stamen, it is having only two parts: the outermost protective layer called as epidermis, and the inner meristematic tissue. Itna hi, kuch nahi, or kuch nahi, because it is so small, very young, like a small baby. Now, this small baby, that means young stamen, if it wanted to produce the pollen grains, it need to undergo differentiation. It need to have a little more complex structure. How? Let us see. At the at the maturation, onset of maturation, this immature stamen, it is undergoing a differentiation. By the differentiation of this meristematic tissue of the young stamen, it results in the formation of the archisporial cell. Now I need to tell you one important point. Till now, whatever the points I told only for your understanding. Just I wanted to know, I wanted to tell you how the staminate is formed, and then I wanted to continue how this particular pollen grains will be formed. That is the reason why I took this topic also. But openly I'm telling you this, just listen it and leave it. Don't read it. Why? Our syllabus, I'm talking about NEET syllabus, it started from here. So from this corner, from this particular point, you need to read. And above this particular point, you just listen it, and then you understand, and then just leave it. But remember it, because how the staminate is formed, that is important. But it is not important for your NEET syllabus. That you need to understand clearly. Now this archisporial cell, it undergoes division, and it it is called as periclinal division. Hope you know what is the meaning of periclinal division. Basically cell division, it is of two types. The first division, it is called as periclinal division, and the next division, it is called as anticlinal division. Periclinal division means the division which results in the formation of two cells. Obviously two cells will be formed from the parent cell, and these cells are arranged one above other, then that is called as a periclinal division. Anticlinal division means the parent cell, it undergoes a division, and by the division two cells are formed as usually, but these cells are present side by side, one after other. That means one just next to that another one, then that is called as anticlinal division. But this archisporial cell, it is undergoing the periclinal division. That means the cells formed are one above other. The upper cell, it is called as parietal cell, and the inner cell, it is called as sporogenous cell. Now this parietal cell, it undergoes continuous division to produce this anther wall. And sporogenous cell, I'll just explain, but let me first finish what is anther wall. Now this anther wall, it is having four layers. The first layer, it is called as epidermis, and the second one is endothecium, and the third one is middle layer, and the fourth one, it is tapetum. Let us take one after one so that you will understand clearly. The first layer is epidermis. Epidermis is a single layered. Unilayered means what? Single layered. Uni means single, layered, that means only one layer. Now it is the outermost layer, and obviously every one of us know epidermis, it is all meant for protection. So epidermis, it is giving protection. And the second layer, it is called as endothecium. Endothecium is again a single layered, and it is just present below the epidermis. Now one point I'll tell you, it is very important point. You have to remember this point. What is that? Please listen. The outer wall of this particular cell is thin, whereas the inner wall and radial wall, so inside and also the sides of these particular cells, it will be thick. Why? It is due to the presence of two things. The first one is cellulose fibers, and the next one is callose bands. Because of the callose bands and also cellulose fibers, the inner portion of this cell and also the side portions of this particular cell, it will be thick, whereas the outer portion, outer walls of this cell, it will be thin. Hope you understand. So inner layers, they consisting of what? Inner layer, it is having cellulose fiber and also callose bands, and the outer wall, it will be that means outer side, it will be very thin, and this is due to the presence of this particular cellulose fibers and also callose bands. But here you remember, I told you one point you need to remember. Yes, I am coming to that point. The callose bands and also these particular cellulose fibers, they are absent at a region, and that region, it is called as a stomium. This stomium, it is the region of the endothecium where the cellulose fibers and also callose bands are completely absent, and that is the region where the dehiscence, that means the breakage of this particular endothecium, takes place. And because of the breakage of this endothecium, it results in the pollen dehiscence. Pollen dehiscence means, the process of releasing of the pollen grains out of the anther is called as pollen dehiscence. And pollen dehiscence, if it pollen dehiscence means the dispersal of the pollen grains. If it wanted to take, the endothecium need to break, and the stomium is a region where the endothecium will break. And the reason is, stomium is a thin area where this callose bands and also cellulose fibers are absent. Hope you understand. But still, if you get any doubt, I'm always for you. You can immediately post me whenever I see. Immediately I'll give you the answer. Now let us take one more important point. Already I told one important point. Now the second important point of the endothecium is, these cells are hygroscopic in nature. Very important question. Why? They have capacity to absorb the atmospheric moisture content. So the tissue which is having the capacity to absorb the atmospheric moisture, and that particular tissue or that particular cell, it is called as hygroscopic nature, or hygroscopic cell, or hygroscopic tissue. Whereas once the pollen grains are fully matured, then the pollen grains need to be liberated. They have to thrown out. How? Now what happens, these pollen grains, when they fully attains maturity, this endothecium slowly starts absorbing the moisture content. When it is absorbing the moisture, it results in the increase in the volume of the endothecium. Now when the endothelial, all the endothelial cells, they are increasing in their size, yet a point will come. What happens, this endothecium cannot be able to hold its strength. That's why it breaks. For example, I'll tell you, you are you putting the books in the bag. You keep on adding the books. Let us take just example के लिए मैं बोल रहे हैं, that bag capacity is of 10 books. Now you put 11 books, चलेगा, no problem, it can be able to resist. Now you keep 13 books, चलेगा, somehow it will tolerate. Now you keep 15-20 books, then what happens? Obviously the sides of this particular bag, it breaks, है ना? Like that, when this all the cells, they starts increasing in the volume, slowly the layer, it starts breaking. And the breaking region, it is nothing but a stomium. That means this is a region where the breakage it starts, and at once it breaks, and it results in the liberation of this particular pollen grains out at once. And that is a function of this endothecium. So hope you all must have understood endothecium. It helps in the pollen dehiscence. Now the third layer, students, hope you, I think I am explaining you clearly, but still if you have any doubts, you can WhatsApp me. Now the next layer, it is nothing but middle layer. And middle layer, it is of 2 to 3 layers. Please don't get confused. Epidermis single layered, endothecium single layered, even the fourth layer, that is a tapetum, that is also single layered. That means out of 4 layers, 3 are single layered. Only the middle layers are middle layer is what I call 2 to 3 layered in its thickness. Now it is a 2 to 3 layered, as I just mentioned now, and it is made up of this parenchyma tissue, and these are short lived. That means once the tapetum is formed, immediately the middle layers will be disappeared. That is the reason why it is also called as epimeral tissue. Hope you know what is the meaning of epimeral tissue. The tissue which is having a very short span, they are called as epimeral tissue. Or even the plants which are having very short span, they are also called as epimeral plants. Hope you know this, all these points. You hope you all know this. And now this epimeral tissue means it is a short-lived tissue. It once the tapetum is formed, obviously it gets disappeared. Now what is a function of the middle layer? For example, epidermis, the function is protection. Endothecium, it helps in pollen dehiscence. Like that, middle layer also must be having some function. What is that function? It helps in the storage of the food material, nutritive material. It will be stored in the middle layer. Now the next layer, and of course the last layer of the anther wall, that is nothing but tapetum. Now tapetum, it is the innermost layer, and of course it is also single layered, and it is a nutritive layer. How this gets nutrients? See, the tapetum, it absorbs the nutrients from the middle layer, and that is how the tapetum it becomes nutritive layer. And once all the nutrients are sucked or absorbed by the tapetum, these middle layers they become empty, and they starts disappearing till the tapetum form. Middle layers are present, and once the tapetum formed and it absorbed the nutrients, this middle layer it disappears. Now, one important point you need to understand in tapetum, what is that? Uh, Tapetum is like a normal tissue at the time of origin. That means when it is formed, when it took birth, it is like a normal, as usual cell, like any other cell. All the cells are diploid, and tapetum is also diploid. But eventually, slowly, this cell it undergoes endomitosis. Due to endomitosis, the normal diploid tapetum during course of time it becomes polyploid. This is a very important point you need to remember. You 100% need to remember. At the time of birth, tapetal cells are diploid, but after some time, due to endomitosis, they becomes polyploid. Now sometimes it undergoes binucleated also, but they never become bicelled. Tapetum, one cell, it remains as one cell only throughout its lifespan. Why? Only karyokinesis takes place, but never cytokinesis. That's why sometimes it becomes binucleated, or sometimes it becomes, I'm sorry, or mostly it becomes what, polyploid, and sometimes it becomes what, binucleated. Now, as I this point I need not to tell you, clearly I told it absorbs the nutrients from middle layer, and that is how it becomes nutritious, and that nutrients it will be taken by the microspore mother cells or microspores. Now, this tapetum, the first function, as you came to know, that is nutrition, and the second one is it helps in the secretion of several enzymes. That is a callase enzyme, which helps in the digestion of callose. I yesterday also few students asked this question. What is this callose? Callose means it is a kind of sticky substance which holds this particular four pollen grains like a mass, which is called as pollen tetrad. And the pollen tetrad it should not remain like that. They need to get separated. That's why this enzyme that is called as callase enzyme, it is produced by this particular tapetal layer, so that this pollen tetrad, which is surrounded by the callose substance, it gets dissolved, and this pollen tetrad it becomes four individual pollen grains, and that particular enzyme it is produced obviously by tapetum, and also it helps in the what I call several hormones which helps in the recognition of the own type of the stigma, so which is called as pollen pistil interaction. Now the pollen grain it gets disappears in a mature anther. That means, are you observing it while I'm explaining? Are you observing it? On maturity, only the epidermis and endothelium only remains. That means out of four, only two are remaining, and two are disappearing. Middle layer, soon after tapetum formation, it disappears, and once the um, uh, this what do you call anther it becomes matured, and this tapetum also gets disappears. So out of four layers, only two are present, endothelium and epidermis, and remaining two are getting disappeared. Whereas this tapetum it also produces the Ubish bodies. That means Ubish bodies means these are the bodies are sac-like structures which are filled up with sporopollenin. And sporopollenin, as every one of us know, it is the world's toughest material. Why it is called as world's toughest material? Because so far, I don't know about future, no one know actually. Till now there is no enzyme to digest that particular sporopollenin. That's why till now sporopollenin is the hardest substance. That means what I wanted to tell you is, till now there is no enzyme found to digest this particular sporopollen. And of course, rest of the story you actually know. Sporopollen formation it results it is the one which helps the pollen to maintain its longevity of the lifespan, and also once it die also it is only the responsible one for the formation of the pollen fossils also. And of course, the tapetum is having some other functions like this tapetum, it produces the pollen kit and also tryptophan substances, and also the proteins that are produced by this tapetum helps to recognize this same kind of this flower. That means some rose flowers are there, it need to identify the rose pollen grains, need to identify the rose flowers only, then only it can be able to germinate, otherwise it can never be able to germinate. So that particular recognition capacity, it is also done by the secretions that are released by the tapetum, and that is called as pollen pistil interaction. Now let us take the types of tapetum. Types of tapetum, first one it is called as amoeboid tapetum, and second one it is called as glandular or secretory tapetum. And amoeboid tapetum is the most primitive type, it is a like a very primitive type. And what is its job, it absorbs the food material from the middle layer, and soon after that it disappears. So the middle layer immediately it gets disappeared. And in the beginning all this particular food material will be stored by the tapetum, and the tapetum cells convert the absorbed food into special food granules, and that special food granules that is called as protoplast bodies. Understanding it? That means amoeboid tapetum, it is all meant for the storage of the food material and converting them in form of a special bodies, special granules called as protoplast bodies. Okay, and this type of this amoeboid tapetum can be seen in typha and alisma, something like that, and tridescantia, and these are the examples for this particular amoeboid tapetum. Whereas this glandular or secretory tapetum, it is the most developed, and most of the angiosperms they consisting of this particular glandular or secretory tapetum. Whereas this do not degenerate so quickly, it even remains after the after few time after the maturation of this particular microsporangia. Now this tapetum, see this amoeboid tapetum, the storage for the food material, but this glandular tapetum, it helps in the formation of a special granules, and this special granules it is called as a pro-ubish bodies, and this pro-ubish bodies slowly they convert into ubish bodies, and this ubish bodies it is having the sporopollenin. Hope you understanding it. Here amoeboid only food material, whereas this glandular it is what you call ubish bodies, that means sporopollenin. And this sporopollenin, it is slowly it forms the outermost layer of this pollen grain, which is called as exine. Whereas this tapetum helps in the transfer of food and storage of the food and formation of sporopollen and also pollen kit. Hope you know many times whenever I get the chance, even revision time also, if you remember, I told you what is pollen kit. Pollen kit is a sticky substance which is produced by this particular tapetum, so that this pollen grain can be able to stick to the bodies of this particular insects, because insect body it is made up of chitin. If on chitin this pollen grains are dropped, this sporopollen exine it come in contact with that particular body of the insect, so it roll off and it gets wasted, and it has to attach to the body. Chitin is so smooth, that's why when the pollen grain it falls on it, it immediately slips. But how the insect can be able to carry this particular pollen grains, that's why this pollen kit makes that particular pollen grains little sticky, so that the insects can be able to carry the pollen grain from one flower to another flower. Hope you understand what is pollen kit. Now, yes, now pollen sacs. There are four pollen sacs, because the anther it is a four tetraangular structure. The tetraangular structure it is called as microsporangia. The four corners are there. That's why four pollen sacs are present, and this consisting of this particular microspores, and they undergoes a meiotic division to form microspore mother cells. And this I'll scroll up and I'll explain it a little more detailed way. Now I'm scrolling up. Yes. Now, the sporogenous tissue which are present in the pollen sac, they transforms themselves into microspore mother cells, and because of this microspore mother cell formation, it enables the reduction division. So this microspore mother cells, they undergoes a reduction. See here, I clearly mentioned microspore mother cell. It is called as MMCs. That means microspore mother cell. Of course, you can also call it as pollen mother cells, because microspore, pollen grain, both are same. You people don't get confused, and they undergoes reduction division. It results in the formation of microspore tetrad, and microspore tetrad, that means four pollen grains stay together by a substance called as callose, and this callus they get dissolved, and it results in the formation of four separate pollen grains. And yesterday, one student, to be frank, one student, around 2 3 students, they asked for the formation of mature pollen grain, one meiotic and one mitotic division is required, but for the formation of mature male gamete and mature male gametophyte, one meiotic and two mitotic divisions are required. And students, I really did not understand. I gave a so clear answer, but still students, they kept on discussing on this question for so longer time. I really don't know where I made mistake. At least this video clarify you. Now, see, I'll tell you. This is a sporogenous cell. Now the sporogenous cell, it is converting themselves into this microspore mother cell. Idhar tak samajh mein hai na? Now this microspore mother cell, it is undergoing reduction division, because microspore mother cell is diploid, but pollen grains must be haploid. That's why it is undergoing one reduction division. See, one reduction division, it results in the formation of four haploid microspore tetrad, and this is a reason why pollen grains are formed. So for the formation of the pollen grain, one meiotic division and one mitotic division is enough. But for the formation of male gametes, because if you take the pollen grain, again the pollen grain, it consisting of what you call one vegetative cell, and the second one is generative cell. In the generative cell, it is again undergoing one more mitotic division to produce two male gametes. That means already here one division for pollen grain formation, that means vegetative cell and generative cell, one mitosis, and again the generative cell, it is undergoing one more division to produce two male gametes. That means 1 plus 1 2. So here already, as I clearly mentioned, one meiosis. So for the formation of one mature male gametophyte, you have to see one meiotic division and two mitotic divisions. Hope at least now you are clear. If you still don't understand, you please ask me. And one more question, I have seen one more question. Some student, of course I did not see the name. If I see also, hope you all know that I don't remember the names actually. One student, sir, then in what I call two celled, because you as every one of us know, if you want I'll show it also, I clearly show it, then you will clearly understand. One second, just me scroll it up, and then I'll show you now. 60% is angiosperms, the pollen grain shed in two celled stages. Yes, and in remaining, that means 40%, you can be able to see they are shed up in they shed up in what I call three celled stages. And this is also I don't know, they got confused. See, yes, they are getting shed up in two celled stages, but once the pollen tube it is formed, that means once the pollen grain reaches onto the stigma, obviously what happens, the intine, it gets stimulated by the vegetative cell, and then it is producing the pollen tube. And after pollen tube formation, the generative cell, it enters into that and undergoes one mitotic division, and that's all also you need to take it now. Hope you are understanding my point. So these are the two questions where we have taken long discussion on this. I am answering and you are questioning, I am answering, you are questioning. Of course, hope at least this video it clarify your doubt. Now, let us take this pollen grain structure. Hope you understand what is pollen grain formation, that means microsporogenesis. Now, let us take pollen grain structure. Pollen grain, It is having a pollen wall and cell and two cells. Pollen wall, it is also called as pollen derm or sporoderm, you can call anything. And this pollen grain, again, it is made up of outer exine and inner intine, and this was proposed by, and of course proved, proposed and also proved by Fritsch. Don't remember, no one will ask you, just a piece of information I am giving. Whereas let us take pollen valme, the first layer, that is called as exine, which is made up of sporopollenin. And that sporopollenin, it is highly resistant, and this is how this pollen grain can be able to tolerate many adverse conditions. And also once if it die, the fossil formation also, it is due to the sporopollenin only. Whereas sporopollenin, that composition or that arrangement, it is not even, it is having minute gaps, and that minute gaps are that uneven thickened, that means the less thickened, or I'm sorry, I'm telling you something wrong. The region where the sporopollenin is not there, and that region, it is called as germ pore. From this germ pore only, what happens, this pollen tube, it is emerging out, coming out. And here also one question, and this question, one teacher also asked me, I don't want to take the name actually, I usually don't like to take the names. Sporopollen is very what do you call, hard substance, and no enzyme is found, no enzyme is so far discovered to digest this particular sporopollen. But we are taking sporopollen, that means pollen grains, in form of the pollen capsules and also pollen syrup, sir. Then how it get digested? Dear, please try to understand what am I trying to tell. Here, sporopollenin, it never get digested. One point. Point number one, sporopollenin, it never get digested. Then how you can be able to utilize the nutritive qualities of that pollen grains? See, the pollen grain, sporopollenin, when it is entering into our body may, it consisting of HCl. It is a very alkaline, 1.8 pH. See, 1.8, highly acidic, so that the structure, it gets ruptured. The sporopollenin, just it undergoes mechanical breakdown. For example, sir, one student asked, I don't want to take the name, he may feel bad. No, sir. The digestive juices enters into the pollen grain, and it digests, and so that all these particular what I call nutritive substances, they come out and leaving this particular pollen grain empty. ये लॉजिक क्या है? मुझे तो बिल्कुल समझ में नहीं आया. Why, sir? Why you did not understand? You may ask. अरे यार, inside you can see intine. Are intine, it is also made up of cellulose, na? Of course, pectin and cellulose. Are in our body cellulose will be formed, cellulase it is formed. Are हमारा body में appendix, it is vestigial, यार. When vestigial appendix it is present, how it can be able, vermiform appendix, and how it can be able to produce the enzyme called cellulase? That means in our body, intine also cannot be able to get digested, and also the exine also cannot be able to digest. It need mechanical rupture. Please try to understand. And some students, they strongly believe what they know, they believe it like blindly. So it's not correct there. This is a point where you need to understand the things instead of reading the things. So please try to understand. I think, hope I did not disappoint your intentions. I just wanted to tell you, this is how the things takes place. But I never mean to hurt you. So let us come to the point. So sporopollenin, it just gets breakdown, so that even exine also, they just gets breakdown, and then it can be able to utilize the nutritive qualities of that particular pollen grain. Hope you understand. Now, the next layer, it is nothing. I hope you understand, but still you have doubt, you can ask me. Now, the intine, it is the innermost layer, as I clearly mentioned. And it is not like a discontinuous layer like exine. It is a happily, it is a continuous layer, and it helps in the process of the formation of the pollen tube by the guidance of the vegetative cell. That is the reason why the vegetative cell, it is also called as tube cell. Now, the vegetative cell is a nutritive cell, which helps in the storage of lot of storage material. And also the generative cell, it behaves like a male gamete once it is undergoing the mitosis. Okay, this is all about the structure of the pollen grain. So hope of course I know, see, while I'm talking only I'll know, I know what I thought. I went a little fast. This is a topic, hope the students who attended this class, I have taken 3 days to explain this particular topic because it is a lengthy topic basically, but I am taking revision, so I know I went fast, but still I am thinking, so I am little clear only. But still, if you don't understand, don't hesitate to ask me to make one more video. Still, I'll make. But my final motto and my final expectation is you need to understand. That is the only point. And you can ask me twice also. 2 times I'll make video on the same topic also if you want. If you understand, very very good. If you don't understand, you ask me. Then a little more slowly I'll make one more video. Thank you so much. And some students, they asked the video lecture on embryogenesis also. Now after half an hour or 1 hour, I'll start making that video also. And for that also, I need to make the notes. Today, this all notes I prepared it, and then I am making this video. And embryogenesis ke liye I need to make notes now, and then I'll make the video lecture. It takes 1 more hour, so by the time you can read it.